So welcome to this talk on a Moneses uniflora propagation research project. The project is made up of propagation trials in both the RBGE nursery and in the field, as well as lab research, with results expected in 2026. It's funded in part by the Cairngorms National Park Authority, with some help from Plant Life, who are working with Moneses uniflora, among other plant species, in the Cairngorms and beyond. Plant Life's conservation work is really interesting, so please check that out if you can. I'll give you a bit of a background, including the ecology, early life cycle, and interactions of the plant to put the project into context, before explaining what's been done this year to help us understand and conserve our Scottish Moneses populations, as well as what we might hope to do in the future. Moneses uniflora is a rare, UK red-listed, partially mycoheterophic, evergreen herbaceous perennial in the family Ericaceae, subfamily Pyraloideae, reproducing clonally or by seed. Quite a mouthful. It's a very niche specific plant though, that has evolved some complex and highly fascinating interactions, so it gets away with it. With Moneses populations declining in Scotland, this project aims to establish an ex situ collection of the nationally scarce plant at RBGE for research and conservation. This small plant can be found growing rhizomatically through the humid bryophyte layer on our pine forest floors. It clumps together in patches of rosettes, rooting in the humus, often hidden by brash. Previously a plant of Caledonian pine woods, it grows alongside the mosses Hylocomium splendens and Hylocoma delphis triquitris. Current Moneses populations are often coastal, with most found in plantation stands. As already mentioned, Moneses is a partial mycoheterotroph, relying on fungi for the first stages of its life cycle. Moneses population decline could indicate biodiversity loss in our woodland soil microbiome, with forestry practices such as clear cutting and agricultural fertilizer runoff reducing soil mycorrhizae, leading to a reduction in germination interactions. Reduction in soil biodiversity may not be the only cause of population decline though. Overcollection in the 20th century, plant geeks do tend to love its intricacies, as well as climate change, could be at fault. With only 6 Scottish populations remaining, conservation is essential to maintain ecological diversity in a habitat that's already limited due to being nutrient poor. Moneses seed is dust-like with an undifferentiated embryo and limited energy reserves, quite similar to a terrestrial orchid. You can see how small it is in the petri dish on the left. The seed requires external nutrients to complete germination, which it obtains from its mycorrhizal associates. Seed probably begins germination processes within 6 months of setting. Establishment includes a seedling subterranean phase, in which the seedling remains underground without creating photosynthesizing parts for an extended and currently unknown period, probably lasting over a year according to one study. You can see a seedling on the right starting to create its green parts. The seedling was subterranean in a pot for at least a year, though possibly longer as I don't know its origin. A Swedish study by Johansson et al. In 2015 found carbon-13 enrichment in subterranean seedlings, so we can assume that there will be some mycoheterotrophy at this stage. So carbon-13 is the mycorrhizal form of carbon, which the fungi has obtained through exchange with an autotrophic plant, usually a tree. Finding it in plant tissues could prove that Moneses is actually taking part in a triangulated nutrient exchange called a tripartite relationship. This takes place between a tree, in our case a pine, mycorrhizae, and a mycoheterotrophic plant. The trees, which create carbon through photosynthesis, can't reach all the essential nutrients and solutes in the soil. Mycorrhizae can reach these essential nutrients, but can't create carbons, which they need to create their hyphal threads. An exchange takes place. Trees give mycorrhizae carbons in exchange for nutrients and solutes. Mycoheterotrophic plants have evolved to intercept this exchange, obtaining some of the carbons which show up in the form of carbon-13. How amazing and wonderful and weird and utterly exciting is this? It makes your head explode. What's really going on beneath the soil? A study by Heintzen et al. In 2015 found Moneses associating with Amphinema basioides, an ectomycorrhizal fungi in the Basidiomycota that's been associated with wood rot, and it was found in 2 Scottish populations. Swedish research suggests that associations might be fluid though, and Moneses probably associates with many different types of mycorrhizae at different life cycle stages. This would make Moneses facultative in its fungal interactions, helping it adapt into new communities. Though evolutionary mechanisms could result in more specific interactions over time. An example of this is a Moneses population in the American Olympic National Forest, which is thought to be partially mycoheterotrophic right into adulthood, something not proven in our Scottish populations. So, due to all this complexity, propagation isn't simple. It requires replication of the associations we've mentioned. Previous attempts have used soil from populations to germinate seed, as well as in vitro propagation, both axonic and symbiotic. This project uses a combination of seed baiting and soil inoculation techniques to propagate Moneses while studying its soil associations. 3 populations were chosen for this study: RSPB Abernethy, Old Granton Wood and Roselle Forest. It will be really interesting to see if there's a correlation between the abundance of mycorrhizal fungi associated with germinating seed and the population numbers. Understanding the soil microbiome could give us an indication as to reasons why some populations are declining. Notice that the population numbers increase the further north they are. Are populations moving northward as a reaction to climate change, looking for cooler areas? To set up the project, seed was collected from Roselle Forest, cleaned and placed back out into 3 populations in mesh packets. The packets are made with a fine mesh, fine enough to keep the seed in, and with just enough space to let fungal hyphae through. These were buried amongst the populations and then left to associate with indigenous mycorrhizae, hopefully kick-starting the germination process. The map shows the seed baiting sites at RSPB Abernethy. 5 treatments containing 25 replicates each. Each replicate is a net bag containing approximately 100 seed. Were planted out in squares among the Moneses populations. You can actually see some of these squares in the previous slides. 2 control squares were planted over 100 meters away to ensure that germination is a result of mycorrhizal interactions. The seed packets will be retrieved in intervals from planting out of 1, 1.5 and 2 years. All packets will be brought back to RBGE and split into 3 separate groups. The first group will be grown on in pots pre-inoculated with soil from the populations, containing juvenile pines and a bryophyte layer to create that microhabitat. The image to your left shows an example of the pots set up in the RBGE nursery. We have rigged up a wee system to maintain humidity while complying with biocontrol measures. The second group will be grown on in vitro for observational study of the Moneses life cycle, and also the seedling subterranean period and the interactions between the mycorrhizae and the plant. And the third will be subject to DNA analysis, obtaining information on the genus and species of the mycorrhizal associates. So we hope that these methods will cover all potential bases, providing the interactions needed for germination, establishment and continued cultivation at RBGE. Further understanding of Moneses interactions will not only shed light on its conservation requirements, but also that of its associates such as the mycorrhizae and pines, as well as other wintergreen species which are also in decline. C13 enrichment, pine root fungal diversity and impacts of climate change on populations over time would all contribute to a better understanding. If anyone has any suggestions refunding for this type of research, please get in touch. I can't stop thinking about what might be happening between mycoheterotrophs and the soil microbiome, so please feel free to enable me. I have left you a little list of light reading to get you started on all things pyraloidy, mycoheterophy, pinewood ecology, and soil biodiversity. It should get you through the darker evenings of winter into spring 2026, when there might be some results to share. So thank you very much for listening, everyone. It's great to have an audience to talk at about the things my loved ones roll their eyes to at home while slowly edging towards the doorway. I do love an email, so please feel free to write if you have any questions or advice. Just expect lots and lots and lots of words back. Thanks very much.